after I tell this story of Corvus, Corvus showed ITK inhibitor Soquelitinib, also known as the CPI-818, proof of concept in atopic dermatitis, and they raised 175 million follow-on after stock soared 2.5-fold within 1 week. So the target ITK belongs to the family of BTK or Tec kinase families, as shown schematically. The top line represents BTK, Tec, and ITK with the plextrin homology, BTK homology, proline-rich domain on the N-terminal, and together BH and PRR are also called the Tec homology domain. And then you have SH3, SH2, and kinase domain. And there, the two Y tyrosine residues are for phosphorylation, and the C481 is the covalent binding site for ibrutinib, which inhibits BTK as well as ITK. And at the bottom, I wanted to point out, previous slide, there is a kinase called RLK. It's not the Tec family, but it also has the structure of proline-rich domain, SH3, SH2, and kinase domain. So next slide. And the company slide came from their January 20 data sharing, and I recommend everyone to watch the webcast by Richard Miller. He is a legend in biotech. He first developed Rituxan, the first approved monoclonal antibody for cancer, and Rituxan now is part of Genentech portfolio. But he developed Rituxan in IDEC, and IDEC was acquired by Biogen, and then now it's, Biogen and IDEC, and Genentech both have a profit split on Rituxan. And in his talk about ITK, he truly grasped why ITK is such an attractive target. As shown here, ITK modulates T cell differentiation into Th2 and Th17 cells. And because of that, their ITK inhibitor, Soquelitinib, blocks Th2 and Th17, so blocks the type 2 inflammation. And so you can see that really blocks the upstream of IL-4, IL-5, and IL-13 from the Th2 pathway, and then IL-17, IL-21, IL-23 for the Th17 pathway. But more interestingly is the recent discovery that ITK also regulates switch from Th17 to Treg. So when you block Th17 differentiation, you actually increase the Treg. And so they report this MOA with their ITK inhibitor as well. Next slide. And that's really, even previous slide, there's also the role of RLK. One more slide, yeah, back here. You see in the Th1, even though ITK is also involved in that, but because of RLK's role, so you really did not touch Th1 and you preferentially blocks the Th2 and the Th17. Next slide. So this really brought back the memory when I worked on ITK more than 30 years ago as a postdoc at UCSF. ITK at that time was cloned, but no knockout, and I was competing with 4 different labs at Harvard, MIT, Berkeley, etc., to try to knock out ITK. And it was known also as TSK or EMT. And I cloned the mouse ITK, CDA, as well as genomic region. And I prefer to use ITK over the other names. And because I published first, so this name got to stay. I'm so happy because it is in parallel with BTK. That was my only reason I wanted it to be known more broadly as ITK instead of EMT or TSK. But this mice one more click, did not have phenotypes. By that, it means no embryonic lethal or other traumatic and obvious phenotypes that got you Cell Nature Science publication and top university appointment. And so I had to look for phenotypes, meaning find the function of ITK through the knockout. And I was lucky to collaborate with three labs whose heads all won Nobel Prize by chance or by their other work, I have to say. Two of them actually after my collaboration with their labs. And this paper, I'm very proud to say, really established the role of ITK and the key functions that Richard Miller mentioned in the previous slide. The failure of Th2 development was reported by us in immunity, and then the RLK and the TLK overlap function but different function was in collaboration with Hal Vamos and his fellow Pam Schwartzberger. This was a science paper. So really, this pioneer work established ITK as an attractive target because no phenotype also means it is a quite safe molecule and potentially very good as a drug target. Next slide. And they have a good molecule, as you can see in this kinome. Soquelitinib only inhibits ITK, and while ibrutinib inhibits BTK, ITK, and many other kinases. Some people were even suspecting that ibrutinib's role could be because it's target ITK as a cross-reactive inhibitor. So some second generation ibrutinib, such as Beijing's, was truly focused on only targeting BTK, and that's great. And I want to say is the pursuit by Corvus, led by Richard Miller, was really prominent because he worked on ibrutinib when he was at Pharmaceuticals. He developed the first protocol for the first in-human trial of ibrutinib with Stanford Hem-Onc fellow, Dr. Dan Pollier, and Dan actually presents the first clinical poster of ibrutinib at ASH. You can read all that story from this book, For Blood and Money. And I'm proud to say Dr. Dan Pollier was also a lead investigator for Immune-Onc, my current company's lead AML asset, because he, like previous presenter said, AML has such unmet needs, and he switched to focus on AML. Next slide. So this new data of January 20 disclosure was focused on cohort 4, but they also did some update of cohorts 1, 2, and 3. These three cohorts' initial data as the primary endpoint was shorter, only 4 weeks of treatment, have been disclosed earlier, and all eyes were on cohort 4, which enrolled additional 24 patients, 1-to-1 randomization, and treated with the dose of cohort 3, 200 milligram twice every day. And the primary endpoint is obviously safety. This is a phase 1. Secondary endpoint is the efficacy, percentage change in EASI. EASI is 75, EASI 90, and IGA 0 or 1. So this data drove the stock rally. Next slide. And I want to show you the cohort 4 data here. It's also treated longer for 8 weeks. And so study day goes to day 56. As you can see, compared to placebo, the purple line showed the reduction of EASI change from baseline. And more importantly is off treatment. After this study, the patients stopped using the drug. The efficacy is maintained, and they attribute that to the activation and the increased Treg, and that is really disease modifying. Next slide. So here is the summary of EASI 75, 90, and IGA 0 to 1 for cohort 1 to 3 at day 28, because they only treated for 4 weeks, and for cohort 4 at day 56 because of 8 weeks treatment. So on the left side, you can see with increased dose, if you just focus on EASI 75, the dark blue line, you see dose-dependent increase in efficacy, and that's further enhanced when you treat longer for 8 weeks. And even subtracting the placebo, it's very prominent. Next slide. And that's where you compare with the current available treatment in atopic dermatitis. Although, it is a cross-trial comparison, but the ITK inhibitor Soquelitinib is very competitive. If you look at the Dupixent from Sanofi and Regeneron, IL-4 receptor alpha that inhibits IL-4 and IL-13 signaling. This was the time when there were many more naive patients, and they got the efficacy 36%. This is placebo subtracted. And Lebrikizumab, which was later acquired by Lilly, IL-13 is 43% of patients reaching EASI 75. And then IL-31 is mainly targeting itch. And it turns out this ITK inhibitor also inhibits itch. So it's really targeting the type 2 inflammation very, very well. And it's almost comparable to JAK inhibitor, which has a black box labeling, while the ITK inhibitor so far in their phase 1 has no obvious safety signal. It seems to be a very safe drug. So this is really impressive. And especially they work very well even in atopic dermatitis patients with prior systemic therapies, including Dupi, JAK-I, steroids, and even clinical trial investigational new drugs. Next slide. So this is the summary that really presents ITK as a very attractive target, targeting Th2 cytokines, IL-4, IL-5, 13, and 31, target Th17, IL-17, IL-21, IL-22, and ILC, as well as increased Treg. And you can see this is a very satisfying. And I was anxious actually before their data readout. And for what I know about ITK, I believe it's an attractive target. But Genentech, during the time I was there, published a very prominent paper in Science Signaling about inhibiting ITK's kinase activity is not sufficient for Th2 inflammation. And obviously, the scientist, the lead scientist, who is currently a CSO of a biotech company, a strong immunologist, and I really appreciate that he actually came to my office, even though I was not part of the research, and he consulted with me about the ITK work. To my satisfaction, he recreated ITK knockout mice, and it reproduced all the discovery I made, but he claimed that just inhibiting kinase was not enough. So I was advising people to do ITK degrader. And Corvus, Richard Miller's presentation said that their inhibitor actually also causes ITK degradation, somehow because the half-life of ITK is shorter. So, but this presentation of the target, many Wall Street analysts call it a validated target now, even though the drug is yet to go to phase 2 and phase 3, and attract and will attract many fast followers. The good thing is Corvus is already developing an ITK degrader. Next slide. Really want to summarize the business aspects of Corvus. I admire Richard Miller, who happens to be the husband of Sandra Honing, who used to be the chief medical officer of Genentech, Roche. And both of them are hematology oncologists and faculty at Stanford. So we knew that connection. And Richard was really instrumental in developing Rituxan and ibrutinib, and now he's pioneering ITK inhibitor. I'm very proud of his accomplishment. And they also developed more like a joint venture, more like a wholly owned subsidiary in the other direction in China, which is Heji Yaoye, Angel Pharmaceuticals. They allow Heji Yaoye to do a lot of Angel Pharma, to do a lot of exploratory studies in China, including higher dose of ITK inhibitors. But this is really practically controlled by Corvus, including its CEO, CFO, and co-founder, board member, Pete Thompson from OBMed. Next slide. I want to summarize my two topics and reflecting my observation. First, in-class drug development against novel targets is always very hard. Like the CCR4 story of RAPT, it's tried immuno-oncology, then tried immunology, and in both cases failed. And I'm glad to see ITK is now making some inroad. And both companies pivoted from immuno-oncology, IO, to immunology and inflammation, I&I, switched the CCR4 inhibitor from IO to atopic dermatitis and failed there, and then bought a half-life extended anti-IgE from China, but stayed on the theme of being an immunology company. Corvus distanced itself from adenosine A2A receptor inhibitor and anti-CD73 antibody in IO, which were also the first two targets of ARCUS, right? And ARCUS and RAPT are related, as Shichang mentioned. I don't want to spend more time on that. But now Corvus focused on ITK inhibitor for T-cell lymphoma, which is a targeted therapy. It's not an IO. So I want to emphasize the difference as well. IO becomes an old, tired name, unfortunately, because of the setback. So they switched to targeted oncology and I&I. And you do this while you still have money or can get investors to give you money. They both are public companies. And Corvus actually was very gutsy. They risked the ongoing clinical trials not being able to finish because they didn't have enough money to complete them, but instead still diverted some of that money to invest in new programs. And many companies cannot do that because you really don't know whether you get new money. And RAPT, their pivot really catered to investors interested in validated target, proven market size, potentially best-in-class molecules, and from China, that's an added advantage because such molecules are quite abundant in China. And they got additional pipe money in parallel to this licensing because the investors knew they were doing such licensing, and they were happy to put in more money to the company. They both leverage, especially RAPT, leverage the data from China even after the deal. RAPT, as a company, did not enroll a single patient outside of China or after their licensing. And Corvus went the other way, which is an innovative way. They invited some Chinese investors to fund their China company. So they really direct everything that Angel Pharmaceutical is doing through US. And finally, a successful exit wipes out all previous failures and bad decisions. Now you are called an experienced management team. And that's all.
Yeah, that's great. I think we should have, this topic is so fascinating. I would like to have more live Q&A. So I will pick some questions from the audience. One question is, does ITK have scaffold function? Some papers seems to say that. Do you agree?
Yeah, so that's why that's the Science signaling paper, a very powerful paper from a well-respected and trusted scientist from Genentech, and said from his work, and people use his two compound, the ITK kinase inhibitor, in many preclinical studies. So when I did a knockout, you wiped out the protein, right? And when he did the comparison of knockout versus kinase inhibition, and came to the conclusion that inhibiting kinase activity does not prevent Th2 or type 2 inflammation, people naturally would come to that conclusion. So that's why I'm so happy to see the Corvus ITK actually works, at least so far in phase 1.
Okay, great. Next question is, it's surprising to this question that the ITK trial did not see any placebo effect. Usually placebo is very strong in atopic dermatitis trial. Do you have any comments?
No, 20%. The one slide is placebo adjusted when you compare with other competing or marketed molecule. But when you look at their trial data, it's EASI 75, placebo had 20%, which is an overestimate actually, because they have two patients reach the EASI 75 out of 10 evaluated at day 56, because there were two patients who did not show up at day 56. And these two patients never reached the EASI 75 in placebo group. So if you do conservatively, it's 2 out of 10 placebo effect, but in true to the trial would be 2 out of 12, which would be 17% placebo effect.
Yeah. One final question. The other question you can address in the Q&A tab. But I'm curious, when you mentioned Beijing, has their compound is very specific for BTK without hitting ITK. But intuitively, hitting both could not be hurt. So you're implying that be more specific with BTK could be beneficial than ibrutinib hitting both BTK and ITK. Could you comment on that?
Yes, later people realize BTK is not just limited to B cells. It's also found in myeloid cells, and in multiple sclerosis, it's in microglia cells. So that's why you do and you can hit BTK specifically and target B cell and the myeloid cells. So that's an advantage. And by not targeting ITK and the BMX or many other kinases, as you saw in the kinome tree, you improve the safety as well. So that's why Beijing was able to do a head-to-head trial with ibrutinib and beat it in a particular indication in oncology.
Also,